Yeah.
Can CBD help with arresting the cell cycle to combat cancer?
So here's a small intro to hemp. A fun fact, hemp can hold 2 times the weight of steel and is 6 times more malleable than steel. One of the main components of hemp is CBD. In ancient China, actually, hemp was used for food, pottery, and many other daily necessities. It's been linked to halting tumor growth, and it's associated with processes such as autophagy and apoptosis. Another fun fact is that George Washington actually had a hemp farm. Now, if we go to the cell cycle of apoptosis, the cell cycle is dependent on cyclin CDK complexes. Specifically, CDK2 and cyclin E complex promote entry into S phase. Apoptosis is programmed cell death, which involves caspase proteases that become activated when cleaved in response to apoptotic stimuli.
So in terms of current research being done on this topic, we found an interesting article discussing the specific correlation between CBD when it's inserted into cells and the effect it has on the cell cycle and apoptosis. In the past, researchers have known that CBD does cause these things, but this paper specifically looks at what proteins are being affected and the level of regulation and efficacy of those proteins. And they actually do this by using some of the methods that we've talked about in class. So what they did was they looked at human gastric cancer cells in vivo, and they used techniques like Western blots and cell colony formation assays, as well as a few other methods to get all their results.
So they first looked at cell viability and saw that treatment of cells with CBD reduced cell proliferation in a concentration-dependent manner, and this effect was greater after 48 hours compared to 24 hours. And upon further examination, they found that CBD reduced CDK2 and cyclin E expression at higher concentrations, such as 40 micrograms per ml. There are thinner, fainter bands for those proteins, so there is less of that protein present. And here, actin was used as a loading control with consistent levels expressed to ensure that an equal amount of the samples was loaded. For apoptosis, they observed that the core apoptosis proteins were present and they were cleaved from at higher concentrations of CBD, which is when they would be active. For instance, there's a very thick band at 40 micrograms per mil for cleaved caspase 9. The results show that the cell cycle is stopped from going into the S phase because CDK2 and cyclin E levels are reduced, which promote entry into S phase, and the apoptosis pathway is activated as caspase proteins are expressed to initiate apoptosis. The future of the field focuses on using CBD therapeutically for cancer. However, it still has to be determined if similar effects will be found on other human cell lines and if there is a concentration that can harm healthy cells. It appears to be an exciting new cancer research treatment.